Last week, we learned how DNA is replicated during interphase to pass down the genetic information to new cells. But now we're going to learn a little bit about how the DNA actually codes for proteins. These are the worker molecules that are necessary for virtually every activity in your body. From enzymes to receptors in the cell membrane, from antibodies that are defending your body against foreign invaders, to hemoglobin in your blood that carries oxygen throughout your body. The code for what these proteins look like is contained on the DNA. More specifically, it's contained within a gene that is on the DNA. Genes are specific sequences of bases, those A's, T's, C's, and G's, that encode instructions on how to make proteins. Genes comprise about 2% of the human genome. The average gene is approximately 3,000 bases, and the largest human gene is about 2.4 million bases. There's estimated to be between 20,000 and 30,000 genes in the human genome. So, how do we go from a gene to a protein? First off, DNA is only found in the nucleus, and proteins are only made outside the nucleus on the ribosomes, which are located in the cytoplasm. Houston, I think we might have a problem. A molecular cousin of DNA, called RNA, or ribonucleic acid, is used to carry these messages. And today we're going to meet 3 different types of RNA during the protein synthesis process. First things first, let's learn a little bit more about RNA before we even begin to talk about how proteins are formed. RNA is almost like DNA except for 3 differences. 1, instead of a deoxyribose sugar, it contains just a ribose sugar on the sugar phosphate backbone, hence the name difference. So ribonucleic acid for the ribose sugar and deoxyribonucleic acid for the deoxyribose. RNA sugar just has 1 less oxygen atom. Second off, one of the nitrogenous bases is different. I know, you thought you had it all down. DNA contains guanine, which pairs with cytosine, as well as adenine, which pairs with thymine, right? However, in RNA, the thymine is replaced with a cool base called uracil. Uracil pairs again with adenine. So in RNA, you have G that pairs with C and A that pairs with U. And finally, RNA is single-stranded, whereas DNA is double-stranded. Remember, DNA is a double helix. There's 2 strands. All right, so let's get back to making proteins. Protein synthesis occurs in 2 steps. First, you have transcription, where the genetic information from a strand of DNA is copied into a strand of mRNA, or messenger RNA. That's the first of the 3 types of RNA that we're going to talk about. And then secondly, after transcription, you have translation, where that mRNA, with the help of the ribosome, forms a chain of amino acids. And when those chains those amino acids are linked together, they form a protein. These events are coined the central dogma of molecular biology. You probably learned a little bit about this last year. So during transcription, the DNA is going to unzip, just like it does when you're copying DNA to DNA, like we learned last week. Can you guys remember the name of the enzyme that unzips the DNA? Yeah, it's called a helicase. So after that, the free-floating nucleotides are going to find their complementary bases with the help of an enzyme called RNA polymerase. But remember, we're making mRNA, so one of these bases is going to be different. Instead of a T, we're going to have a U. And then finally, a new sugar phosphate backbone is going to be formed with a ribose sugar. And your new mRNA is going to separate from that DNA strand, and you're going to have a single-stranded mRNA. This whole process took place in the nucleus, and the mRNA strand will leave the nucleus through one of the nuclear pores. All right, let's try it. What RNA strand will be made from the following DNA sequence? Take your time and write it down on your paper. So during step 2, the translation process, that mRNA is decoded. It will be changed into or translated into an amino acid sequence. So remember, way back when we studied macromolecules, you learned that the building blocks of proteins are amino acids. This translation process takes place in the ribosome. Ribosome is also known as rRNA. So remember, ribosomes are actually made up of a combination of RNA and proteins and can either be free-floating in the cytoplasm or bound to the endoplasmic reticulum. We call this the rough ER. The mRNA is going to park itself right at the ribosome. But we've run into yet another problem. There are only 4 RNA bases, but there are actually 20 different amino acids. So in order to determine the protein the mRNA is going to make, we have to divide the sequence into 3 bases. These base sections are known as codons. And since each 3-letter combination codes for an amino acid, the cell will need to figure out what an amino acid matches up with each codon. So at the ribosome, also known as rRNA, we have our last type of RNA. It's the tRNA, which interprets the sequence from the genetic code corresponding to which amino acid. The individual transfer RNAs, or tRNA molecules, come along and read the mRNA base sequences 3 at a time. The tRNA with the correct anticodon, or the opposite sequence as the codon, will match up with the mRNA. tRNAs act like a little transfer bus. Each tRNA carries its own amino acid passenger. So remember that there are 20 different amino acids to choose from. Once the tRNA drops off its amino acid passenger, it leaves the ribosome. Each amino acid then gets deposited at the ribosome and is joined to its neighbor amino acid by a peptide bond. A peptide bond is just a covalent bond. Remember, covalent bonds share electrons, and this holds the amino acid group of one amino acid to the carboxylic group of another amino acid. The ribosome moves over 3 more bases on the mRNA and a new sequence, or to a new codon, and then another tRNA arrives that matches that new sequence and brings another corresponding amino acid. This process keeps on repeating itself, and before you know it, you've got a polypeptide, then a protein. So to end that polypeptide chain, you hit a codon or a stop codon on the mRNA that has a matching tRNA that doesn't carry any amino acids. So when this tRNA arrives, the translation process stops and the protein is now terminated or stopped as well. So in summary, the mRNA is made in the nucleus and carries a message from the DNA to the cytoplasm. The tRNA, they transport the amino acids to the mRNA to make a protein, and the rRNA makes up the ribosome, which makes our proteins. So now we have this long strand of amino acids, but in order to be a protein, this string must fold up into a 3-dimensional shape that will give the protein its function. So the next thing we're going to do is understand how proteins fold into these cool shapes. Proteins fold in 4 levels of complexity. So step number 1 is the primary structure. That's simply just the sequence of amino acids that makes up the protein. It only involves those covalent peptide bonds linking each amino acid together. So just a brief note, on average, an average human protein is made up of about 355 amino acids linked together. So folding step number 2 is the secondary structure. There are 2 main types of secondary structure. You've got the alpha helix and the beta sheets. These secondary structures are defined by patterns of hydrogen bonds between the main chain peptide groups that form these regular shapes. And then folding step number 3 is the tertiary structure. This is that 3D structure of a single protein molecule, where those alpha helices and beta sheets are folded into a more compact form, also known as a globule. That's a cool word to say. This tertiary folding is driven by some non-specific hydrophobic interactions, where Certain amino acids hate water, and so they want to go to the inside of the protein. There are other amino acids that have hydrophilic attractions, and they want to be close to the surface of the protein to touch the water in the cytosol that's surrounding the protein. So since there are 20 different amino acids, each one has its own unique chemical makeup, and each amino acid will have a different characteristic. So some will be positive, some will be negative, some will be hydrophobic, others hydrophilic. So they each interact differently to help that protein fold up. Finally, some proteins are made up of more than one subunit. So when there are two or more polypeptides joined together, this is called a quaternary structure, and hemoglobin is actually a great example of that. Finally, you should know that every protein has a particular pattern of folding, and these patterns can be really complex into a really crazy three-dimensional structure that's going to give that protein its function. Proteins can also be unfolded or denatured by things that can break these bonds. We talked a little bit about this when we were talking about enzymes. Things like extreme pH or even high temperatures can cause a protein to unfold and to no longer have these bonds. I hope you guys enjoyed learning about protein synthesis and how proteins are made. We're going to be using this information today in class and also tomorrow to put what you just learned here in this video to practice. All right, have a great day. Bye.